Hello. Uh,
Hello.
Welcome to Science Mission, Hangout from Science Mission. So this is our eighth Hangout. So we have today Dr. Charan Ranganath, Professor of Psychology from University of California, Davis with us. Professor Ranganath has won several fellowships and awards from various universities and organizations. So he won an award from Memory Disorders Research Society. He also won Samuel Sutton Award for Distinguished Scientific Contribution to Human ERPs and Cognition. Dr. Ranganath won Chancellor's Fellow Award from UC Davis and the Young Investigator Award from the Cognitive Neurosciences Society. In 2015, he was one of the 11 researchers in the United States to receive a National Security Science and Engineering Faculty Fellowship. Welcome, Dr. Ranganath.
Thank you.
So can you please tell us about yourself, your education background, that will help people to know more about you?
Yeah. So I was born in, back then they called it Madras, India. Now it's called Chennai. So everything has changed there.
Okay.
But when I was very, very young, we moved to the U.S. and where my dad did his PhD at Brown University.
Okay.
And then we moved to actually Arlington, Texas, right near you.
Okay.
And from there, we moved to California, San Jose, which is where I grew up.
Okay.
I went to college at Berkeley, UC Berkeley. Went to graduate school at Northwestern. And then did postdoctoral research at University of Pennsylvania. And our whole lab moved to Berkeley. So I helped set up the imaging center at Berkeley.
Okay.
And then I got my faculty position at UC Davis back in 2002. And now I'm a full professor here at Davis.
Okay. And like, have you thought about going to the industry before you move to the faculty position? So it's kind of a tricky question. So, but
yeah, no, it's I sometimes wonder about that. So back when I was, you know, kind of getting ready to graduate, that was when the tech boom was taking place in the Bay Area. But I had actually gone into college as an electrical engineering major, which would have been perfect. Except within about a year, I realized that I hated engineering.
Okay.
I'm the kind of person who likes to cause problems, not solve problems. So,
okay.
So I didn't really fit in with that. And, I worked as a temp at some software company 1 year during the summertime. And I just got so bored, I started stealing office supplies. And so
Okay.
I won't name that company so that they can't come back to me. But and I think that was the experience that made me realize I don't want to work for someone else.
That is interesting. Yeah, okay, that's good. Good to know some of your stuff, what you had done previously. Yeah, like, I mean, like, do you have any mentors during your career? So, like, people who supported you in your career, helped you move up the ladder and all those things?
Yeah, well, actually, so, you know, probably the biggest academic mentor I had was my graduate advisor, Dr. Ken Paller. And so he had moved to the university while I was actually working on a completely different topic. I was doing research on brain physiology and depression. And but I just took his class when he started at Northwestern, and it was a class on human memory. And I ended up talking to him about an idea for an experiment, and the next thing you know, I was in his lab doing the experiment. For about 6 months to a year, I actually didn't even know if I was in his lab, but eventually he took me into the lab, even though I was not, you know, I was not necessarily admitted as one of his students. And he showed me pretty much all of the basic skills you need to do science and communicate it effectively. mean, he put in a lot of time into mentoring me, and so it was kind of, he was my biggest role model in that sense and
Okay.
mentor.
Okay.
And then I think two other people who obviously played a big role were Mark Desposito, who I did my postdoc with,
Mm-hmm.
and Dr. Marsha Johnson, who was at Princeton at that time. Now she's at Yale. And we had a
Okay.
collaboration. And so she was intellectually a very, important person in my career development.
Okay.
And kind of each of them was contributed in different ways. I think with Ken Paller, the big contribution was showing me how to do science, how to write papers, how to think about science.
Mm-hmm.
And in with Marsha, it was about, kind of building on those skills and also learning more about theories of memory and, how to put together different kinds of phenomena that people have studied in memory.
Okay.
And with Mark, it was about linking what we did in memory to neuroscience research, say in animals, which a lot of psychologists at that time didn't even really look at much.
Okay. Mm-hmm.
And then also we, really Mark was a pioneer in using functional magnetic resonance imaging to
Okay.
study brain function, and that's where I learned that stuff. And one of the things that Mark was very, adamant about was that people in his lab, it wasn't enough just to use the method, but you actually had to understand the method. You had to kind of get your hands under the hood.
Uh-huh.
And, so you had to kind of know how things worked. And that was huge because when we moved to Berkeley,
Mm-hmm.
I was, one of the most senior people who moved with Mark,
Okay.
There were others too, but it was one of those things where I couldn't ask other people for that much help. We really had to figure things out for ourselves.
Yeah.
And that was a hard time, but I really learned a lot from it. And
Okay.
I think that taught me some of the most important lessons about running my own research program.
Oh, okay. That's interesting. And what are the challenges do you face each day, like other than what you are doing?
Well, I think the challenges that I face, let's see, there's so many. Balancing family and work is probably the biggest one, and making time to,
Mm-hmm.
sort of have, things that are outside, kind of like things that I do to kind of relieve stress and relax.
Mm-hmm.
It's hard to balance all that stuff. I think at work, one of the big problems that you have is that people don't realize this because they think, oh, professor, you just teach and you do science or whatever.
Mm-hmm.
A huge chunk of my time is just taking up, doing meetings with people, handling paperwork.
Mm-hmm.
Every research grant you get, you have to write progress reports and respond to emails from the people there, but also just email itself. I could get if I went on vacation for 2 days, I'd come back to at least 200 emails.
Yeah.
It's so those things are. I think the biggest thing that is difficult is the amount of non-teaching, non-research time that I spend,
Mm-hmm.
and also it's. I think there's some pressure to, at the same time, I've got a very big research group under me, and
Mm-hmm.
it's very important to me that I'm personally involved in that research, and I'm not just a manager,
Yeah.
because that's everything I wanted to avoid doing was just, being some part of this big machine,
Mm-hmm.
And so it's very important for me to do that. But it's just hard to make time for everyone. And so that's, in general, time management is the big challenge.
Mm-hmm. Mm-hmm. Okay. And can you talk about some of your lab people and the project, some of the things they are doing?
Yeah. Actually, well, before I just realized the other big challenge is keeping everything funded.
Yeah.
You probably hear that a lot. But basically, when I was in grad school, when I was an undergrad, you never get trained for how to manage a budget and manage a staff. And,
Uh-huh.
I've at times been managing, a yearly budget of like around $1 million. Right now, it's more like $500,000.
Mm-hmm.
And what I didn't realize until I started doing it when I ran my own lab is that it's a constant juggling act, because you are thinking right now of like, okay, so I have these many projects that I've committed to do for my research grant. So I need this many people to do the research. And the thing is that within, anywhere from, within 4 years, you're already realizing that the project should be almost done. Meanwhile, you had graduate students start in year 3 or year 4, or you had postdocs start then, and they're not ready to move on. And so somebody has to pay for them.
Yeah.
And so you get up in this endless cycle of writing a grant to fund people that you have, then you get it, then you need to hire more people, but then you need to get more funds to keep them going after, because there's these two competing goals. One of the goals is to, sort of mentor people in your lab and have them do good research, but then you need the funds to keep them going, and the funds aren't always in sync with the people.
Yeah. That is interesting. Yeah, it's a challenging role.
Yes, yeah, and the funding agencies expect you to spend the money they give you each year, but the actual demands on the money vary from year to year.
Year to year.
I think that's a big part of it too.
Okay. And I think we should learn more about what your people are doing, what your lab is doing. So can you please go ahead and do your presentation?
Oh, you want me to give the presentation now?
Yeah. Is it possible?
Sure.
Can you?
Let me just share this. Go. And can you see this okay?
Yeah.
Okay. So first, this is an old picture of my lab. Actually, some of the people in this picture are gone, and we've added some people since then. But most of the people there are still in the lab. So I just want to kind of acknowledge them, because anytime I say we are doing something, it's really they're doing something. So also we have many collaborators I've listed here. We've received many sources of funding. One of the most interesting sources has been through this Proposition 63, which gave allocated money towards mental health services. And so we've been funded to do some really exciting research, which I'll tell you about from that program.
Okay.
So the title of my presentation is How Memory Works and Can We Improve It? Now, those are big questions, and I'm not going to answer either of them conclusively. In fact, if there's any memory researchers watching, I suggest you turn this off, because you'll probably otherwise get mad and then email me all these things. But I'm going to try to present a simple version of some of the work we do. But if anybody out there has questions, you're welcome to hit me up on Twitter or email me or whatever, and I'm happy to help you with the details and answer any questions or concerns you might have.
Okay.
So, my lab is called the Dynamic Memory Lab. One of my first students named the lab, so I've just kept it ever since. But, basically when we started off, we were really interested in understanding just how the brain is organized and understanding how memory is organized. And so understanding how the different processes that you do to remember a past event are related to different brain systems. And so after a while, sometime after my first midlife crisis, I decided I should really use this knowledge to understand memory in different patient populations. And so that's been a big part of our research. And then around my second midlife crisis, I decided we should actually stop talking about how bad memory is in many of these patient groups, but try to work on making it better. And so we've got some exciting new directions there. And so I'll tell you a little bit about all three, but I think I'll emphasize more about memory disorders and memory enhancement, because I think people in the general public probably are more interested in those lines of work.
Okay.
So, to study the brain, first of all, I think it's important to use the methods that are available to ask different kinds of questions. And I think this is something I've been very, kind of adamant about in my research. And it's fairly, there's not that many people who use all of the approaches that we use, although, there's quite a few out there. One of the techniques which you're probably very familiar with is functional magnetic resonance imaging. And so this is a technique where you put people in an MRI scanner and you use imaging sequences that are sensitive to changes in blood oxygenation.
Okay.
And that's correlated kind of indirectly with brain activity. And so you can see what areas are showing increases in activity when people remember a past event.
Dr. Ranganath, I don't see the change in the slide. Are you changing the slides?
My PowerPoint seems to be stuck. Oh geez, my screen seems to be stuck. Everything seems to be stuck.
Yeah.
Oh no. Okay, there we got it. So let's see. Okay, I'm gonna try this again.
Okay.
Sorry, this is my first time trying to do this on a Google Hangout. So
okay.
Okay, I think somehow PowerPoint has quit on me. I'm going to stop sharing for a moment.
Okay.
I think I'm just going to quit this and start it again. Good old Microsoft.
Mm-hmm.
Well, anyway, while I'm waiting for this to start up, one of the techniques that we're using is kind of something that's really fundamentally changed how we study the brain, which is not just looking at what areas light up when you are trying to remember something, but also to, okay, here we go, but also to try to actually, can you see any of this, by the way?
No.
Okay, so I have to turn on screen sharing then.
Yep.
Okay, so one of the techniques we're using is using information in the brain activity patterns to basically read out what kind of information someone's remembering at a given time. And so what we can do is we can use this technique to study specific brain areas and say, when you remember an event, is this area telling you something about where the event took place or who was in it, what was going on? And so this has been kind of a major area of our research recently.
Okay.
Another method we use is we study patients with focal brain damage. So here's a picture of an MRI from somebody who had a temporal lobectomy for epilepsy. And so these patients are very helpful to say, okay, if you actually had damage in a particular brain region, how does that affect your memory? We also use a technique called electroencephalography. So here's a picture of the former quarterback of the UC Davis football team who's working in our lab. And you can see he has a cap. Can you see my mouse arrow here?
Yeah.
Okay, so he's wearing this cap and it has electrodes embedded in the cap. And so the electrodes actually contact the person's head, and electrical signals that emanate from the brain can be picked up by these electrodes. And so you can analyze them for different kinds of activity that happens at different frequencies. And this is called brain oscillations. And so, for instance, here you can see a very long oscillation that has a has a frequency of 6 hertz. And understanding these oscillations can tell us a lot about the language that the brain areas use to communicate with each other. Here's a picture of an MRI scan from a person who has epilepsy, and before they do these kinds of surgeries, they actually have to stick electrodes in the brain to figure out where's the region that they want to take out. And so we actually could do research with these patients because, let's say they don't need to take the hippocampus out in this patient. We can use recordings from these areas to tell us about what the hippocampus would do when people are learning new events. Then another technique we're using is brain stimulation. So in our lab, we're using, just like we can record electrical signals from the brain, we can actually transmit electrical signals through the scalp and have it affect activity in different brain areas. And so here, for instance, is a model of the effects of the electrical activity on a particular part of the brain for the subject. So, when I teach my classes on memory, I always start off by asking people, why is it important to understand memory? Because when I was in school, I couldn't really make any progress on learning anything if I didn't understand why it was important. I think for many people who might see this webcast, the first thing that comes to mind is, how memory is affected in different kinds of diseases, and Alzheimer's is definitely the first one that comes to mind for sure. I imagine that of people who are going to be, viewing this, probably 2/3 of those people will know someone, fairly like a first-degree relative or something of that nature, who actually, experienced dementia, maybe Alzheimer's disease. And almost anyone who'd be watching this would know of somebody in their life who ended up with dementia or Alzheimer's disease. And so that's obviously important. But there's other disorders too. For instance, we're hearing a lot about traumatic brain injury, especially in soldiers returning back from war, and for people who've experienced concussions in the football field, for instance. And actually, it's not that much known about precisely how concussions, for instance, affect memory, in the sense of it's obviously bad, but people don't know whether if you have a concussion, should you stay out for a long time, or should you just keep getting concussions, and when is enough enough, but a lot of our research is actually focused on understanding psychiatric disorders too, which people don't think of as being affecting memory. But in disorders like schizophrenia and depression, actually the cognitive deficits, the memory deficits you see in these patients are what really are debilitating and what's very hard to treat with medication. Meanwhile, in post-traumatic stress disorder, you have the opposite problem, where it's not a matter of failing to remember, but it's actually remembering events that are, just intrusive memories of events that these patients can't stop. And also it's a disorder of generalizing memories, because it's like they recover these memories at times and places which don't match up with the event itself. And so, some of the research on PTSD is concerning how patients generalize memories across different kinds of contexts. Anyone who's got a kid usually becomes very interested in memory, because you want to see how your child's memory will develop over time and relate it to their education, and of course aging. I mean, nobody above the age of 20 will say that their memory keeps getting better and better as they get older, or if they do, I'd love to study those people. And so we want to understand the trajectory of memory during healthy aging, as well as age-related memory problems. I think for all of us, we've experienced the frustration of not remembering where we parked our car, or not remembering where we put our keys. And I think, understanding memory gives you a very clear idea about the limitations of memory. So the first studies of memory were done by a scientist named Hermann Ebbinghaus. And basically what he showed was forgetting happens very quickly. So he would learn this kind of boring, meaningless information. And within about 20 minutes, he would basically have forgotten just a little, about 40% of that information. And within, essentially one day, he would have forgotten 70% of that information. Now, interestingly, whatever stuck around after 2 days was pretty much going to be around, for a long period of time thereafter. But what it tells you is that a huge majority of the events in a particular day are already lost by the end of the day. And so, it's important to study memory to understand what's the difference between those memories that stick around and those memories that you end up losing. And that's been a big component of our research. So
are you moving the slide? I don't see the slides getting moved again.
It's not moving?
Yeah.
Which is the last one you saw?
The research methods.
Oh, that's it? Oh,
Yeah.
no. Okay. Well, I apologize for all these technical glitches. Try turning it off and turning it on again.
Mhm.
Okay, let's try this one more time.
Okay.
So let me go here. Can you see it now?
Yeah, I can see. Yeah.
How about this?
To understand memory.
Okay. So this is what I was trying to show you before about this forgetting curve that you can see here. And what it shows is that over time you forget, but also that forgetting curve is very steep over the first 2 days. And so what we're interested in is figuring out what makes the memories that you lose here different from the memories that stick around over this long period of time.
Slide again doesn't move at all.
I don't know what to do about
Oh, okay,
this.
now how soon we forget? Okay, now it moved.
Can you see this?
Yeah.
Aging?
How soon we forget?
Oh no.
Memory
Okay.
gets,
Well, how
memory
about
gets
now?
worse. Memory gets worse with aging. Yeah.
Yes. So this is these are graphs showing how memory changes with aging using very simple measures. And what they show is that across a group of subjects, people who are older tend to have worse memory. No surprise. What you see here, though, on the right is a longitudinal study of different people. And what you find is the degree to which memory changes with aging varies dramatically across individuals, with some people showing these very steep declines, as you can see here, and some people showing virtually no changes or maybe even slight improvements, although I think this is more of just a statistical issue. But what's interesting is that if you follow the brain areas, some brain areas that are critical for memory, like the prefrontal cortex, which you can see here, and the hippocampus, which you can see here, the volumes of both of these areas change significantly with aging. And some of these, you can see, for instance, for the prefrontal cortex, that the decline parallels the decline in memory pretty nicely, actually. And in the hippocampus, you can also see these losses, but what happens is these losses tend to be really steep after the age of 50 or 60. And so again, if you look at this part of the curve, it again actually parallels what you see in these cross-sectional studies or the overall average that you get in longitudinal studies. Can you see this slide?
Yeah, yeah.
Okay, so we study a particular kind of memory called episodic memory. And just to give you an idea of what episodic memory is, it's essentially a memory for an event that allows you to answer the questions about who was there, what happened, when it happened, where it happened, and how it happened. Oh wait, I have a typo in this slide. I gotta fix this. There you go. And so, for instance, this is a photo that was taken of me when I had this interview in my office, and you could see all of the science stuff in the background. This is a picture that was taken of me when I was in graduate school and I was playing in a band, in an underground band at night. So you could see this person was a lot younger, thinner, had more hair, was cooler maybe than this person. But these are each associated with different places and times and situations. And when I look back at this event, I could differentiate it from this event, even though they both involved me and they might have involved some other common elements, because of this information about the context, for instance. And one of the points in our research and other people's research has shown, in fact, this is probably one of the biggest findings to come up in the last 20 years of memory research, is there's a fundamental difference between the who and what information of a memory and the when, where, and how of a memory. So in particular, what we see is there's a whole stream of brain regions, whole networks of brain regions that process and really develop information about who or what was in a memory.
Mhm.
And this information culminates in an area called perirhinal cortex. Can you see that on your slide?
No, I think you need to point to the on the left side. You have those different slides. Just click on that, so that will help to bring it to the.
Okay, let's try that. Can you see this now?
Now it is basically like episodic memory. So
Yeah.
the brain regions. The next slide.
Okay, so let's try making this even bigger. Can you see that okay?
Yeah.
Okay, that's still not quite right.
Yeah. I think the previous one was okay. Yeah.
Okay. So again, I apologize to the viewers for this problem, but oh, actually, if I shrink it, can you still see just this?
Yeah, just the slide only I can see. Only
Okay, perfect. So you guys are going to lose some of the animation. I apologize for that. So what I was saying before is that there's a whole network of brain regions that processes information about who or what you're experiencing during an event. And that information is put together in this area called the perirhinal cortex. And so we tend to talk about that who or what information as being kind of, we have a nerdy term, which is we would call them items. But information about the when, where, and how of a memory is processed in a very different stream of brain regions. And that stream culminates in the parahippocampal cortex. We call that kind of information context, because it's not about what happened, or it's not about what was there or who was there, but it's about time, place, and situation. Okay?
Okay.
And this information gets hooked up together in the hippocampus. And so people think of the hippocampus as being, you probably heard about the hippocampus as an area that's very important for memory.
Yep.
But the reason why it's important is not because it's storing all of our memories per se, but it's really associating the brain areas that that are storing different parts of a memory and allowing them to be put together, so that the next time I go back to Chicago and I see the street where my band had played, I'll be able to remember, you know, my experience of having played at that club, you know, something like over 20 years ago.
Mm-hmm.
So let me just get rid of this. And yeah, okay. So, you know, some of our basic science, for instance, has been being able to decode patterns of activity in the hippocampus, for instance, and seeing how memories are organized there. And so what we can do is, when people are remembering different things, we can find patterns of activity across different little subregions of the hippocampus. And so that's shown here with a cube that just has different colors, with the colors corresponding to how much activity there is. And so then we can say, is there anything reliable that we can calculate from the pattern of information when people are remembering particular things? So for instance, if we remember two events that occurred at a similar time, do we see a higher, you know, do we see some reliable signal in the hippocampus? And when we see memories that occurred at different times, we can see that there's much less of a reliable signal. So something about the hippocampus tells us about when an event took place, but it also tells us about what took place at that time. And so one of the interesting things we've found, and this is just kind of a graph illustrating that, is that different brain regions will have access to different kinds of information. So the parahippocampal cortex will have this information about time and space, and the perirhinal cortex will have this information about objects, for instance, in our world. And the hippocampus really tells us information about what you saw and when you saw it, and puts those together. And what's nice about that is it tells us that the hippocampus is organizing memories in a highly specific way. So if you imagine you're trying to do a search for a file in your computer, and that file has a particular file name and it has a particular file type, right?
Mm-hmm.
And if you do a search for just all Word documents, you might be able to find it. If you remember part of the name of the file, you might be able to find it. But if you could search for the file, let's say if you put in the file name and then you could get out the specific document type that's associated with that file name, that would give you exactly what you need to open up the file, right?
Yep.
And so basically what we're seeing is that different parts of the brain have different filing systems. Some areas in the cortex allow you to generalize across memories that share certain information. So just like you could open up a window and access all of your Microsoft Word documents, for instance, different brain regions can access and generalize across memories that occurred at a particular time or place, or involving particular people or things. But the hippocampus plays a unique role in allowing you to file different memories according to both what happened and where it happened, or what happened and when it happened.
Okay.
Does that make sense?
Yeah.
And if I'm not making any sense at any time, just feel free to interrupt.
Yeah, sure.
So we don't believe that memories are localized in a single brain area, though. And one of the things we've learned in the past 20 years is that the brain is organized into different networks, right?
Yep.
So on one hand, you've probably heard the term of 6 degrees of separation, that essentially if you went through all of my social networks, you could eventually find friends of friends of friends who somehow have had, connections to Kevin Bacon, the actor.
Mm-hmm.
And while that's true, for the most part, our lives are actually structured into a few particular social networks, right?
Yep.
So for instance, you have a family network, you might have a work network, and you might have a friend network.
Mm-hmm.
And those networks can be often fairly separated, except for a few people who share, in both of those networks,
Mm-hmm.
Likewise, if you are driving around the Texas area, you're surely going to be, spending time on the freeways.
Yep.
And some freeways will connect up a lot of different areas. Let's say, one freeway might connect up, some major cities. But to get to a particular city in Dallas, you might have to cross over between one freeway and another freeway.
Yep.
And so just as these freeways have a particular territory and particular places that they connect, the brain's white matter pathways are basically pathways that connect up particular sets of brain regions.
Mm-hmm.
And that's what's shown here in this diffusion tensor imaging picture. Now, we can actually look at areas of the medial temporal lobe in the hippocampus and the peritoneal cortex, and what we find is that each of these regions in the medial temporal lobes actually connects up with different networks in the brain. And so this is kind of an inflated picture of the brain, and what this is showing is that the parahippocampal cortex, for instance, connects up with a different network than, say, the perirontal cortex does.
Mm-hmm.
And so part of what we've been doing is kind of moving beyond these particular brain regions to the networks that these brain regions interact with. And so, based on our research and other people's research in animals and in humans, in, people with particular disorders and in healthy people, we put together a model that basically says that these different pathways are allowing you to unpack information in memory and use it in very different ways. And so we have one pathway that allows us to take information about the time and place of an event and put it together into information about essentially a general, kind of a mental model for what kinds of events take place. So for instance, if you are remembering some event that took place in a restaurant, you can automatically retrieve the kinds of things that happen in a particular kind of restaurant, right?
Mm-hmm. Mm-hmm.
So if you go to a sit-down restaurant, you can immediately predict before you even walk in where, that you're going to be seated by somebody. You can have a waiter hand you a menu. You don't pay until the end of the meal, right?
Yeah.
And so these kind of mental models allow you to kind of retrieve little bits of information and then construct a story around that. And that's kind of how we think memory works, is that you get little bits and pieces of details,
Mm-hmm.
but then you put a story around it that allows you to get the gist of the event. And without that story, the memory would be really meaningless,
Yep. Yep.
And another way we get information from memory is we can use it to make inferences about particular people and things. So for instance, your experiences with a particular person might allow you to extract information about whether that person can be trusted, whether that person has a good sense of humor, whether that person cooks good food.
Mm-hmm.
And that information is very useful, again, in helping you make decisions in the future. And we think that's associated with this more anterior temporal network. And so these anterior temporal networks and the posterior medial network, which I'm showing here, oh no, this is another mistake I have in my slide. So I'm going to fix this right now before it becomes, sorry about that. This will seem like such an amateur operation to your viewers. So these actual networks that we can see functionally with fMRI map on to particular white matter pathways, which are like those freeways in the brain that I was telling you about. So one of the major discoveries of the past decade or so has come from the Alzheimer's community, where what they've shown is that these diseases like Alzheimer's disease are neither localized to a particular brain region, nor in the early stages, nor is it completely, the brain damage completely spread across the brain. And in fact, what you see if you look at the atrophy in the brain in Alzheimer's patients, it actually tracks this posterior medial brain system very nicely, right? So you see this here?
Yeah.
And you can see the correspondence between the areas that show brain damage in Alzheimer's disease. So this is a network that's identified in people who are healthy. But what we can see is that that network is particularly targeted by Alzheimer's disease. Now, what's interesting is people talk a lot about the hippocampus in Alzheimer's disease, but there's other disorders that affect the hippocampus too. So, for instance, there's a disorder that's much less well known called semantic dementia. And it's a disorder that affects the temporal lobes and the frontal lobes. And what you can see is in semantic dementia, you get big effects on this anterior temporal system. But that posterior medial system is largely spared in the early phases of the disease, right? And so what people have come to realize is that the symptoms of these disorders, because Alzheimer's disease affects your memory for particular places and particular events early on in the disorder, right? So patients with Alzheimer's disease, the early symptoms are, difficulty remembering events, difficulty remembering how to get to new places, etc. But people with semantic dementia start to lose knowledge about, things and people in their lives. So for instance, they would have trouble kind of differentiating between a horse versus a zebra in terms of their meaning. And so what people have come to realize is that understanding the brain networks that we see in healthy people is hugely important, because they give us ideas for understanding how different diseases and disorders affect the brain. Because they don't happen willy-nilly in the beginning, they actually progress along a particular pathway. So just like, certain serial killers might work along a particular freeway and kill people along every exit on the freeway, these disorders are going through these white matter pathways and just wiping out different areas as they progress through the pathway. Sorry, I guess that was a little morbid for your audience. Are you still there?
Yeah.
Okay, good. So it's a little hard to do these things without seeing people. So
okay.
But where am I now? So essentially, we're starting to use our understanding of these brain networks to not only look at degenerative diseases, but also to understand the effects of different health conditions on the brain. So for instance, a big one, it's actually a huge one, it has personal significance to me because this affected my grandfather, who eventually died because of vascular dementia. It's understanding the effects of cerebrovascular disease. What I'm showing you here is a picture of a typical middle-aged older person's brain, and it's using a sequence that's designed to detect damage in the white matter. And you can see, for instance, this bright spots are areas where the white matter has been affected near the prefrontal cortex, right?
Yeah.
And so this is very, very common in even healthy older people. And what we think it reflects in part is tiny little vascular disturbances, like essentially tiny strokes that are happening in the microvasculature, and they're killing off. They lead to damage to the white matter pathways. And so what's interesting is that we know, for instance, that the prefrontal cortex is critical for retrieving information about past events and using it to make decisions.
Mm-hmm.
And what we see is the more damage people have to these white matter pathways, the worse their memory performance is, the worse their working memory performance is, and the less they're able to activate the prefrontal cortex. So even in these people who you were to look at just a raw image of the prefrontal cortex and you say, oh, their brains look totally normal to me,
Yep.
they still won't be able to use the prefrontal cortex because it's disconnected from the areas in the brain that are holding information in memory.
Okay.
And so, this is particularly relevant because we have an epidemic of diabetes that's happening right now, especially in California.
Mm-hmm.
And diabetes dramatically increases the degree of white matter damage that you see in the brain. Likewise, hypertension, it contributes to these kind of white matter disorders, and things like high cholesterol, et cetera. And so these are fairly treatable health conditions, and they can have effects that are different from the typical Alzheimer's profile, but end up causing even more severe deficits in the long run.
Mm-hmm.
And so it's important to understand these kinds of deficits because we can do something about this, unlike Alzheimer's disease, which we don't have that many options for yet.
Okay.
I think I'll skip this work on schizophrenia because I'm probably running a little behind. How much more time do I have?
Yeah, you have another 15-20 minutes.
Okay, let me skip this one and move on.
Okay.
One of the things that I like to do is I like to collect products that claim to improve your memory.
Mm-hmm.
So, Sadashava, do you remember seeing this anywhere in the store?
Oh, yeah.
So this is a chocolate drink, and they claim that it's going to enhance people's memory. I bought it in India.
Mm-hmm.
And so I actually tried this. It was okay, I guess. It wasn't what I would choose to drink, nor did it improve my memory.
Okay.
But it did have Choco Babam chips in it, so I couldn't complain that much. This is a thing, I actually haven't tried this, but they call it neuro-NZT, and it's supposed to be a cognitive enhancer.
Uh-huh.
So if you've ever seen the movie Limitless, they're calling it NZT to try to imply that somehow if you take this, you'll become like the character in Limitless. My favorite one is this drink that I bought called Neurogasm, which is offensive to me both as a neuroscientist and as a person with taste because it tastes terrible.
Okay.
And of course, it didn't induce any kind of neurogasm when I drank it. So we're trying to come up with better ways to improve people's memory. And so one of them is that we're looking at, maybe I'll skip that. I kind of missed a slide somewhere in here. So sometimes people ask me a lot, will doing something in particular improve my memory? There's certain things like ginkgo, fish oil, vitamin B, certain kinds of foods where people think this is really helping my memory.
Mm-hmm.
The research that's out there suggests that at least if you average across all people, the effects of these things tend to be placebo effects.
Mm-hmm.
But if you research the placebo effect could be statistically significant. So if you actually believe these things are helping your memory, maybe they are. So don't stop.
Okay.
There is a little information about blueberries suggesting that blueberries can affect your memory.
Uh-huh.
And, there's things like brain games or action video games or doing certain kind of puzzles. And, there's some evidence suggests that you can practice these things, they can improve certain kinds of tasks.
Mm-hmm.
There's not a whole lot of evidence to suggest how much it generalizes to the real world and how big those effects are.
Mm-hmm.
So I think there's some promise there, but so far we're early days,
Mm-hmm.
But the most promising things, I think the things that have gotten the most positive results are studies looking at aerobic exercise. And those studies have consistently shown that they have positive effects on the hippocampus. They actually increase neurogenesis in the hippocampus.
Mm-hmm.
And they improve cognition and they improve memory. So as far as the brain goes, if you're in good enough health, aerobic exercise is like a no-lose situation. Okay. Another is sleep. Getting a good night's sleep can really help your memory. Mm-hmm. And worse, not getting sleep can really harm your memory. Okay. And so these are two things that are very important. Unfortunately, they're things that I'm guessing many people who are watching this won't do, and I know because I don't do it. I don't get enough sleep, nor do I get enough exercise, but it's hard to do that in modern life. Another, which I'll talk about in a little bit, is the motivation to learn. I think that's very important. There's things that you need to avoid, like chronic stress, hypertension, diabetes, I already talked about. Obviously, it's very hard for us to avoid chronic stress in our daily lives, but to the extent that you can or you can manage it, that's going to be helpful to you, because a lot of the research shows that even though transient stress can often help your cognition and help your memory, if you experience stress for a long period of time, it can have very bad effects.
Okay.
Can you see this now, or is it?
No, that's it. The same slide, one slide.
So one of the things that we're doing to try to see if we can improve memory is to look at what we call neural oscillations. So if you record electrical activity from the brain, what you'll see is you'll tend to see these kinds of waveforms in the activity. And that means that there's a certain particular temporal structure to the brain activity. And in particular, what we see is if we record human EEG, one of the rhythms that we see in these recordings is that you'll see these kinds of waves in activity that I'm pointing at here, which are around 6 hertz, or between 4 to 8 hertz, meaning that they repeat 4 to 8 times per second. So this would be about 1 second of brain activity. And so what's interesting is these same kind of theta waves that we get in human brain activity, also it's been known for a long time that you can record these waves directly from the hippocampus in rats. And so this is a picture of just raw signal from the electrical activity of a rat hippocampus. And you can see it's also got that theta frequency structure to it. In some of our research, we've shown that people who show more theta activity when they're trying to remember an event are better able to remember both the information in the event and when and where and how the event took place.
Okay.
And so we've got a grant right now to see if we can improve this oscillatory activity in people and see if we can enhance memory performance. And so one of the things we've used is just using rhythmic lights and sounds to basically entrain the brain to produce rhythms at the theta frequency. And what I'm showing you here is some preliminary evidence showing that by doing this, we can actually improve people's theta activity, which is this little burst of red that you see here. This means that there's more activity in this 6 hertz window when people are retrieving memory for the context of a past event, when you've been undergone this entrainment procedure. And what we've shown in 2 experiments now is that basically doing this entrainment procedure improves your memory for both what happened and the context in which it happened. And so this is kind of a promising study. Now, the effects you'll see are very small, but they're highly reliable. We're now seeing if we can use electrical stimulation to try to improve memory. And so this is a picture of the device we're using right now. And what you can see is some very preliminary evidence suggesting that we get a small but reliable increase in memory, again, for this kind of ability to remember and associate different things that happen in an event. For people who got brain stimulation targeted at the prefrontal cortex. And this is a figure showing that the people who got the prefrontal stimulation showed an enhancement in theta activity when they were learning about these associations. And the people who got that stimulation show much more of an effect when people are learning these associations than people who got fake stimulation, where they felt like it, but it was sort of a placebo effect.
Okay.
Now, again, these effects are all small. And what we're trying to do is we realize everybody's head is shaped differently. And so what we're trying to do is use MRIs in order to figure out where to position the stimulation most precisely to target the same areas of the prefrontal cortex in each person. Another thing we've been taking advantage of is the effects of motivation on memory. So, you know, we're probably all familiar with the term neurotransmitters, which is the idea that there are these chemical messengers that send signals from one cell to another in the brain. But there's other there's a particular class of neurotransmitters which you'd call modulators, like dopamine, serotonin, norepinephrine. And these transmitters don't necessarily put activity up or down, but they change the pattern of activity in neural circuits. And in particular, they're important for allowing new experiences to stick around in the brain, memories for new experiences to be able to stick.
Mm-hmm.
And so, in particular, dopamine is one of the most interesting neuromodulators because it's important in energizing the brain to kind of seek out seek out a stimulation that's going to be rewarding to you. So, in other words, it's, if you're hungry, it's going to energize you to go get food. Mm-hmm. Or if you are, bored and you want to, you want something to do, that's dopamine kind of signaling you to try to seek out stimulation, for instance. What's interesting is that dopamine, when it's released during a learning event in animals, it affects the connection strength between different cells so that those cells that are involved in a particular event can become more strongly connected over time, mm-hmm. and therefore retain the memory for that event. Now, one of the things that has been shown in studies of rats is that not only does dopamine increase when rats experience rewarding events, but also rewarding events seem to be preferentially active in the hippocampus. So one of the things that people have found, Matt Wilson at MIT was a big pioneer in this area, was he recorded activity from the rat brain while a rat was sleeping. Mm-hmm. And what he found was if you look at when a rat is moving in space, you'll find a particular sequence of cells in the hippocampus that fire according to these different places that the rat has been. And what he found is that during sleep, those cells fire in the same kind of sequence that they did during the learning event, suggesting that the brain is replaying these events during sleep and using that to kind of strengthen the memory by sort of re-experiencing it over and over. Mm-hmm. Now, when this happens, there's these very characteristic patterns of the brain called sharp wave ripples when these replay events occur. What's interesting is that these waves don't just occur during sleep, but even during rest. So if you get bored of my talk and you start zoning out, there's a decent chance that you will also be experiencing these replay events. And during these replay events, even if you're not experiencing it, your brain will be replaying these elements of the past experience. Mm-hmm. Now, what Lauren Frank at UCSF showed was that during these replay events, you're more likely to see a sequence of events that's been associated with reward. So, for instance, if a rat went in a maze and he went in this direction in order to get this reward, and in an earlier trial he went in this direction and didn't get a reward, what mm-hmm. you see is more preferential replay of the sequence of events that got the reward. Okay. And this is shown here that rewarding events were much more likely to be reactivated during one of these sharp wave ripples than mm-hmm. non-rewarded events. So what we took from this is that this is a great way for memories to become solidified in the brain, because just as if you experience something 5 times, you're going to have much more attention for that event than if you experienced it 1 time, right?
Mm-hmm. Yeah.
And so our brains allow us to experience these events even if they happen once, if they were salient and associated with reward. And so what we said is maybe this replay is not just a mechanism for strengthening memories, but prioritizing memories, because I showed you that not all of our memories during a day survive, right? I mean, a huge chunk of these memories get lost. And so one of the things that we found is that, in fact, I should go back and say, one of the reasons why the brain probably does this is because if we remembered everything, there would be so much competition in our brains for between all of these different memories that we would have trouble finding the memory that we need at a particular time, right? Mm-hmm. So just like if you go into your office and your office is a mess, just like my office is a total mess, I'll have trouble finding something.
Yeah.
And on the other hand, if I keep a fairly minimalist office, I'll be able to find whatever I'm looking for because there's no clutter there.
Yeah.
And so what these replay events might do is eliminate the clutter and prioritize the memories that you really need to find. Mm-hmm. And so we've looked at this in humans by having people experience some events
that happened while they're doing something to get high monetary rewards. We paid them a lot. Other events they experienced while they were getting almost nothing in payment for it.
Mm-hmm.
And then what we did was we trained a computer model to read out the patterns of activity in the hippocampus to find differences between activity when people were experiencing these high reward events versus the low reward events. And what we found is that during rest, we see more reactivation of those brain patterns that are associated with high rewards than brain patterns that are associated with low rewards. And this is just during people just laying back, zoning out in the MRI scanner. And it's a lot easier than you think. Even though people think it's loud and scary in an MRI scanner, it's actually, you know, sort of like, it's very easy to fall asleep even. But these people were awake, but just resting and zoning out. Now, what we saw is people who showed more of this reactivation of the rewarding events also showed a memory bias to remember those reactivated events later on. So what we found was that the replay of these rewarding events is a mechanism that allows you to kind of prioritize the things that are important to you and sort of remove the clutter from memory by kind of putting them at a lower priority for reactivation. Another study, which I'll probably just for time purposes, I'll just present this as the last one, is a study that we did on curiosity in memory. And so this is, again, both this study and the last one was done by Matthias Gruber. If you're interested in him, he's got a TEDx talk that's on the internet. You can see he's very sharply dressed for his talk.
Okay.
We got a lot of media exposure too, if you're interested about this event. I think the NIH director's blog and the NPR site have pretty good descriptions of the study.
Okay.
But what Matthias did was he wanted to look at curiosity because, sure, I can pay you to learn information, right? But my guess is people who are, if anybody's watching, if I haven't alienated all the viewers of this webcast by now, and if you're still watching, it's not because somebody's paying you, because I'm certainly not paying you for it. Hopefully you're not paying them for it. It's because they're curious and they want to learn about it. And a lot of our real life learning is motivated by curiosity. So for instance, I don't know if you've experienced this, but I'll often think of something and I'll just do a quick Google search on it. And then it leads me to search for something else. And then 15 minutes later, I'm just deep in this, you know, sort of like Google wormhole where I've completely lost track of how I even got in there, right? And that's not driven because they're paying me to click on these things. It's driven by my own curiosity. And so we wanted to see, does curiosity have a similar effect on the brain to the effects that we see for motivation, you know, money seeking, for instance. And so we thought about it and we said, well, how do you kind of, first of all, you know, get people to be curious? And some research shows that the way you get curious is often when you have some interest in a topic and you know something about a topic. But I ask you a question that leads you to go, wait, I should have known that, and I didn't, right? So if somebody were to tell me something, you know, say, hey, do you know this about memory? And it was something that I didn't know. I would be really curious to find out because I'm supposed to know a lot about memory. For some reason, I didn't know that. So that would motivate me to seek out that knowledge, right? Just like dopamine can motivate you to seek out a reward. And so what we did was we asked people a bunch of questions. And some of them were things that people were interested in, and some of them were things that people weren't. We asked them to rate how interested they were. And what we then did was we present these questions again in the scanner, and we would keep them in suspense for about 10 seconds before we would show them the answer to the question. And what we were interested in was, does this question stimulate curiosity in a way that's very similar to saying, hey, if you do this thing correctly, I'll give you $5. In other words, does it stimulate you to seek out the answer to the question, much in the way that reward motivation would lead you to seek out a reward? And so we can look at brain activity evoked during this question. And in fact, that's what I'll be showing you during the whole rest of this presentation, is just activity that happens when you stimulate people's curiosity. But we also wanted to see, obviously, you're going to remember events better if you're curious about them than if you don't care about them, right?
Mm-hmm.
So obviously people will remember the answers to questions that you're curious about. But what we also said is, if there's some kind of enhanced dopamine activity that's triggered just by the motivation to learn, then maybe even presenting innocuous, unrelated information like a face while people are waiting in suspense will be enhanced. Your memory for that will be enhanced if you were highly curious because of that enhanced dopamine activity. And so just like you might be in suspense if you were watching, the last episode of Breaking Bad or something like that,
Mm-hmm.
and you're waiting through this commercial break to see how it's going to end, and these people pop up these commercials, would you be more able to remember the stuff from those commercials if you were in a lot of degree of suspense? Or like in the Super Bowl, if your team was playing in the Super Bowl, would you remember those ads more if you couldn't wait to find out if your team was going to win?
Yeah.
And so what we found was naturally people are better at remembering the answers to trivia questions they're interested in. And so that worked, right?
Mm-hmm.
But what's interesting is, even though these faces had nothing to do with what they had to learn, when we gave them a surprise memory for the faces, what we see is a slight but very reliable advantage in memory for the faces that are presented when you're in a state of high curiosity. Now, some people show a bigger effect of curiosity than others. This is something I'm going to come back to when we look at the brain activity, because it's hugely important. And as it turns out, almost every study that's looked at motivation and memory finds these very large differences amongst people.
Mm-hmm.
And in some sense, this makes sense because, you and I probably know people in the world who will do anything for a dollar, right?
Yeah.
And other people who just don't care, and they're not very motivated at all by rewards, monetary or otherwise.
Mm-hmm.
And so, there are lots of individual differences here. And so that's why we wanted to look not only what holds up across people, but also the individual differences of brain activity.
Mm-hmm.
So what I'm showing you here is activity that's evoked not when you get the answer to a question, but just when you get the question, okay?
Mm-hmm.
But we looked at activity when people are getting the question, and then we said, what happens in the brain for when people see a question which they later on remember the answer to, versus questions where, soon after they got the answer to that question, they just forgot about it, okay?
Mm-hmm.
Now, what we find is in the area of the brain called the ventral tegmental area, where dopamine is produced and released to other brain areas, what we see, and I should just clarify, we're not actually measuring dopamine. We're measuring activity in a brain area that, has a lot of dopamine in its neurons.
Okay.
But there are also other neurons in this brain area. So we can't say for sure that it's the actual chemical dopamine. What we can say is activity in this region goes up dramatically when people are more curious. And remember, they're not getting the answer. They're not satisfying their curiosity. We're just stimulating their curiosity here.
Mm-hmm.
But the more people say they're curious about the answer, the more activation we see in the dopaminergic midbrain. And when we're looking at the question, not during the answer, but just during the question, the more activity we see in the hippocampus when people are curious, the better people are able to remember the answer to this trivia question. Now, what's interesting about that is we only see this when people are highly curious. When people are not curious, the activity when they see the answer predicts memory for the answer. But there's no ramping up of activity when people are in suspense. And what we show is the more that ramping you get for when you're in high states of curiosity, the better your memory is going to be for those items. And that probably parallels why people show a better memory for these answers when they're in a high state of curiosity.
Mm-hmm.
So remember, we also had these faces here, right?
Yep.
And so we asked then, well, what happens in the brain when people remember these faces? And I told you that the effects were variable across subjects. So if we actually look at the effects across people, what we see is that some people show a very strong memory advantage driven by curiosity. And some people show virtually no advantage of curiosity, right?
Yeah.
What's interesting is during the question, the more people show a curiosity-related brain activation, in the dopaminergic midbrain or in the hippocampus, the more of a curiosity-related benefit we get. So activity in these brain areas is not just predicting learning of the thing that you're motivated to learn, but it creates this state, the state of anticipation, which allows you to better suck in all of this information that happens while you're seeking the answer, right?
Mm-hmm.
And so it's just like, it's very much like that study I was telling you about, where the hippocampus wants to reactivate not only the reward, but the sequence of events that took place in order for you to get to the reward.
Yep.
And likewise, when you're motivated to learn, you not only activate the reward-related brain system, but you're also going to remember the stuff that happened while you were on the way to getting the answer. And so this is interesting because you're involved in the field of media and journalism.
Yep.
And any good journalist will really practice this without even knowing it. They will bring up something that both gets your attention, but it also brings up some kind of spark of curiosity, because they'll tell you, wait, is there some way I can, improve my memory performance? Or is there some way that I could, prevent my house from getting broken into? Or is there something I can do to train my dog? Whatever it is, they get you curious. And then they tell you what they really wanted to tell you. And they don't even necessarily answer the question that you're curious about.
Yeah.
And what our research shows is it actually doesn't make a difference what the answer was. It's the question, and it's really the motivation to learn that's the issue.
Yeah.
And any good teacher will do this too, is they will stimulate kids' curiosity way before they actually teach them what they need to learn.
Mm-hmm.
And I think this is something we could do much more about in education, is not just emphasize the facts and the answers, but really spend a lot of effort emphasizing the question. Yeah. And emphasizing, empowering students to seek out the answers for those questions. And I think that's something, it's a real life implication of our work. And I'm not saying that teachers don't do that. A lot of good teachers do exactly that.
Yeah.
Providing, kind of a substantive explanation for why it works.
Yeah. That's
so we have some other techniques that I won't really tell you about. But for anyone who's interested in our research and you want to learn more, just keep up with what you're doing. We have a website called the Dynamic Memory Lab, dml.ucdavis.edu. And so if you could just press pause on the webcast and you can write this down if you want. We also have a larger group of memory researchers at UC Davis, one of the largest memory groups in the world, actually. And so we have a website linking to all of these researchers as well, so you could learn about other people at Davis too. If you're interested in random bits of humor, social justice, and memory stuff, you can follow me on Twitter. I might rant occasionally, so please don't hold that against me. If you want a more, just updates on the research and find out when we publish new papers, you can check out our lab Facebook site. You could just search Dynamic Memory Lab on Facebook and you'll find us.
Okay.
And finally, I just want to thank you guys for your attention.
Thank you very much.
So maybe I will take this off of screen share now.
Yeah, yeah.
So yeah, is there anything else I can answer for you or anybody else who happens to be watching?
One question I have is, you have talked about a lot of reward and all those things where memory gets consolidated when you sit around or just nap or whatever. What about, have you looked bad side of it? Like here you are talking about the reward, bad kind of like bad events, bad kind of accidents happening or somebody shooting or some bad things happening. So
yeah, absolutely, absolutely. And what's interesting, actually, so let me step back for a moment and just say that if you actually look at memory consolidation at the cellular level, in order to strengthen those synapses and have that effect last, you not only need to have brain activity in those brain regions, but you also will need to have some kind of a neuromodulator released, like dopamine, norepinephrine, serotonin, etc.
Yeah.
And what these chemicals do is they tend to signal salient events. So dopamine, as I said, signals reward and signals, what you need to do to get a reward, what actions you need to take.
Yeah.
Norepinephrine is involved in generating the fight or flight response when you're scared or when something negative happens to you. And so it turns out that if you look at people who look a lot at memory for negative emotional events, they find that those memories can also be enhanced, but they're associated with the pathways that involve noradrenergic areas of the brain and the amygdala.
Okay.
So it's a different mechanism, but similar results. And one of the things that's been shown is it's the arousal that's particularly important. You can actually even have a positive event, but if it's arousing, like going snowboarding or going skydiving or something like that, where you might be doing it because you enjoy it, but there's still this kind of feeling of danger, and that too can lead to enhanced learning and retention of memories. So it's a similar mechanism, but it's different. Another part of the research shows that essentially when you have a stressful event, that's arousing, it can also affect what memories from prior to the stressful event get retained. And so we have some new work showing that in particular, memories that you might have forgotten, if they're followed by a stressful event, those memories are more likely to stick around after.
Stick around, okay.
And so again, it's kind of not only prioritizing the memory for what happens at the time, but what led up to those circumstances. Just like if you were walking down a particular street, you turn a corner and you get mugged, you would not only want to remember that you got mugged, but the sequence of events that led you to get mugged in the first place. So you don't do that again, right?
Okay. Yeah.
Once you turn the corner, it doesn't help you to remember, oh, I got mugged here. It helps you to know not to turn the corner in the first place.
Yeah.
And so another interesting thing we find, and this is and we didn't do this, but other people have done this, like Elizabeth Kensiger and Mara Mather, have shown that even though we feel like when an emotional event takes place, we remember it really vividly. In fact, there's only certain parts of it that we remember very strongly and effectively. There's other parts that aren't affected or are actually kind of maybe even worse for emotion. So for instance, if you pointed a gun at me right now, if you were in front of me and you pointed a gun at me, I would be very likely to remember that you pointed a gun at me. Anything that's focused around that. Yeah. But I'd have a harder time remembering the color of the shoes you were wearing, or the where you came from before you did that sometimes, because those are backgrounded information. Yeah, they're not as important. And so the interesting thing is people will sometimes have a weak or a completely distorted memory for that background information, but they'll feel like they're remembering it vividly. Because just recalling that context gives them this feeling of arousal. And so they'll say, well, because I feel the sense of arousal, I must have remembered this event. And so, this is hugely important for people who are doing testimony in trials, for instance, or people who are being, questioned by the police, because they'll often say something that's very confident and very inaccurate.
Good. Yeah, it's a very interesting topic. I think we have to learn more, I think, in the future about this topic. And there is a lot to learn also, I guess. Like, I think we are just learning a little bit of whatever is happening in the brain, and it is much more complicated than what we thought previously 10, 20 years ago. And I really thank you for taking this time and talking to me and giving that presentation. I really appreciate your time and effort. Thank you so much.
Oh, you're welcome. Thank you for having me. And, I hope people feel free to connect with us after they see this.
Yeah, certainly. So that is one of the things is like people, if they are interested in talking to you or emailing you, they can do that. And you have given the information where they can find you, basically.
Yes.
So again, thank you so much.
Thank you.